Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about enzymes. Now, there are a lot of chemical reactions going on in any cell or any organism, and every single chemical reaction that's going to occur is going to take an enzyme. That's how the cell does those chemical reactions. So, if you look at all the reactions that are going on in a cell, you start to see why enzymes are so important to life. Now, enzymes, like I was saying, they speed up a reaction, they allow the reaction to occur. Heat, or thermal energy, will also speed up reactions, but will do so indiscriminately. In other words, heat will allow every single chemical reaction going on in the cell to occur, and to occur quickly, whereas enzymes are very specific. They'll only allow one reaction to occur, and they'll only allow the specific molecules involved in that reaction to react. That's why the cell prefers enzymes over heat. It allows it to have more control over what's going on. Now, before we get into exactly how that works, there's a couple terms to describe. If you see this picture, you've got the enzyme, which has an active site. Now, the active site is the part that's actually doing the work, that's allowing the reaction to occur. And it's shaped in a way that it will conform to the shape of what's called the substrate. The substrate is the reactant or reactants that will be placed into the active site. When all this combines, it's called the enzyme substrate complex, which is just a word for the unit of the enzyme and the substrate together. Now, in addition to that, when the substrate is in place, it will actually change the shape of the enzyme so that it fits more closely, more tightly, and this is called the induced fit of the enzyme. Now, like I was saying, enzymes speed up reactions. They do so by lowering what's called the activation energy. And I'm just going to show you kind of what it looks like graphically. The activation energy is just an amount of energy that's required to allow the reaction to occur. I'm going to explain those diagrams more in a second. But the ways that an enzyme can lower that activation energy are as follows. The first is that it can distort the substrate into a transition state. In other words, the enzyme is shaped just so that it'll stretch certain bonds. It'll morph the shapes of the molecules just slightly in order to make it more likely to react. The second type is the microenvironment of the active site. This refers to a couple of things. Maybe there's a certain amino acid in the active site that's very acidic, and this creates an environment where the reaction is more likely to occur. Another example might be with polar and nonpolar amino acids, and this all creates maybe a slightly different environment in the active site than in the rest of the cell. Now, the last one is going to be that the active site directly participates in a reaction. This is the least common of the three. Basically what happens is the substrate will actually covalently bond to the enzyme before breaking, and then it'll be released of that covalent bond with the enzyme. The reason this is the least common is because there are just certain problems that can happen that might damage the enzyme, and in order for an enzyme to function properly, it has to emerge from that reaction without being changed. Now, like I was saying, with the activation energy, these are the diagrams of it. If you want a reminder of what free energy is, go back and check out our metabolism video. It's essentially the energy stored within a molecule that can do work. Now, this level right here is the free energy of the reactants, whereas this is the free energy of our products. The distance between them is the delta G, or change in free energy, for that reaction. That's pretty intuitive, but essentially, overall, it changes from this level to this level, so that difference is the delta G. It's a negative delta G, meaning it's a spontaneous process because it's going down from a higher to a lower level. The problem with this reaction is that in order to occur, you have to first add this amount of energy to the reactants. That's called the activation energy, which we mentioned earlier, and it allows the reactants to change into a transition state. Now, the transition state has the bonds within it are slightly weaker. They're more unstable and likely to break and reform. And when they reform those bonds, it'll reform as the products. So you have to add a certain amount of energy in order to get the products to the transition state, and then they'll release energy as they change from the transition state to the products. Now, if you look at this graph, this shows two versions of the same reaction. The red line shows how the reaction would proceed under normal circumstances, and the green line shows how the reaction would proceed in the presence of an enzyme. Now, like I was saying, the enzyme lowers the activation energy. With the enzyme in place, you only need to get this high in order to get to that transition state, rather than all the way up to the regular activation energy. The important thing to notice is that at the end of the reaction, when all is said and done, delta G is the same with or without an enzyme. This is because the G has to be the same for the same products and the same for the same products and the same for the same reactants. That's just how chemistry works, how physics works. Now I'm going to get back into more of this enzyme stuff. There's a property of an enzyme when it becomes saturated, and this happens when there's so much of a substrate that the enzymes are being filled just as quickly as the reaction is occurring. When an enzyme is saturated, like I was saying, it happens at high substrate concentrations. So it happens at high substrate concentrations. When there are so many substrates, the enzyme can't keep up with it. Basically, essentially there's a line of substrates waiting. If you've ever been to maybe getting tickets at the movies, there's a line. The movie booth or whatever is saturated because it cannot get to everyone quick enough. When an enzyme becomes saturated, in order to make the process occur faster, in order for the reaction to occur faster, you have to add more enzyme rather than adding more of the substrate. When the enzyme is not saturated, that's the opposite. Adding more enzyme won't help at all, whereas adding more of that substrate will allow the reaction to occur quicker. Another property of enzymes is that they have an optimal temperature and pH, and this is where they function the best. This is where they can do their job the best. They can make the reaction occur the fastest. Now, there are certain enzymes in your stomach. Now, of course, your stomach is very acidic. It has a very low pH, and those enzymes that are in your stomach are breaking down proteins. They function best at a very low pH. I think a pH of about 2 in your stomach, which is incredibly low. Whereas the enzymes in the rest of your body function at around a neutral pH. I think 7.6 is the pH of your cells. But the rest of those enzymes are optimal at a 7.6 pH. In fact, they're denatured at the acidic conditions of your stomach, which is actually part of the reason your stomach is so acidic. The same is true of temperature. There are certain bacteria that live in hot springs and just like magma pools. I don't know. But their optimal temperature is more like 90 degrees Celsius. Water, that's almost the boiling point of water, which is 100 degrees Celsius. Whereas your cells are only 36 degrees Fahrenheit, or 36 degrees Celsius, in stark contrast with that temperature. And so each of those enzymes, the enzymes in those bacteria and the enzymes in your cells, have a very different optimal temperature. Now, there's a couple more types of compounds that are similar to enzymes. A cofactor is similar to an enzyme that is non-protein. So it's a non-protein helper for enzyme, or So a cofactor will allow those reactions to occur similar to an enzyme, except it's not a protein. That's the important thing. One example of this is the iron in hemoglobin, which is the protein that binds oxygen in your red blood cells. Without the iron, the hemoglobin cannot bind oxygen. It cannot perform its function. But with that cofactor, iron, it can, and so the cofactor is essential to its function. Now, when a cofactor is organic, iron obviously is a metal. In this case, it's an ion of the metal. But when it's organic, it's called a coenzyme. And there are a couple examples of coenzymes when we talk about respiration, so you can go see that that video if you want those examples. Now, enzymes okay. So for the rest of this video, we're going to be talking about different ways that your cells regulate enzymes. The first is through inhibition. Now, there are two types of enzyme inhibitors: competitive inhibitors and non-competitive inhibitors. Now, as the name suggests, a competitive inhibitor competes for the active site. It binds to the active site, taking up the space that otherwise the substrate would be using. In this little example, the orange molecule or compound is the substrate. The green one is the competitive inhibitor, and the blue one is the non-competitive inhibitor, which we'll be looking at next. Basically, the green competitive inhibitor will bind to the enzyme in the active site, taking up the space where the substrate would be otherwise. Now, a non-competitive inhibitor binds to somewhere else on the enzyme, not to the active site, but when it does so, it actually changes the enzyme's shape. If you remember when I talked about the induced fit of an enzyme, this basically works opposite to an induced fit. When that non-competitive inhibitor binds, it changes the shape of an enzyme such that it no longer can perform its function. It no longer can bind to the substrate, and so that's key to a non-competitive inhibitor. Now, there's also a type of regulation called allosteric regulation. And what you have to know for allosteric regulation is that essentially every protein, or almost every protein, has two or more different forms that it will it'll constantly switch back and forth between those two forms, those two different shapes. An allosteric molecule, or an allosteric regulatory molecule, will bind to a site on the protein and stabilize one of the forms. If it stabilizes the active form, that's an allosteric activator. If it stabilizes the inactive form, it's an allosteric inhibitor. This diagram kind of shows you. This is the active site, and you can see right there too. This molecule actually has 4 active sites, or this enzyme has 4 active sites, and the allosteric activator will stabilize it in this position to where the active sites are available. This is the inactive form where those active sites don't work. They're non-functional. And the allosteric inhibitor will stabilize this form and prevent the enzyme from doing that function. One example of an enzyme that does this is an enzyme called phosphofructokinase. Now, you don't need to memorize that, but it's essential in cellular respiration, which we're going to be looking at in the next couple of videos. And it's regulated by ATP and ADP, and even AMP, which is, those 3 are adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. 3 different molecules that are all very similar, but they're regulated, or they regulate phosphofructokinase in order to control the speed at which cellular respiration occurs. And if you know what ATP is, it's essential to metabolism, to making reactions occur. It's actually one of the main products of cellular respiration. Cellular respiration produces ATP, and so those molecules will they allosterically regulate that enzyme. Now, cooperativity is very similar to allosteric regulation. It's actually one type of allosteric regulation, but it uses a substrate itself. Now, if you remember, the activator or inhibitor was binding to somewhere other than the active site. In cooperativity, you have it binding to the active site because it's a substrate. When it does so, it increases all the other active sites' affinities for the substrate. When I give you this example, that'll make sense. The example is hemoglobin. Again, like I mentioned earlier, hemoglobin is in your red blood cells, and it binds oxygen. Now, every time hemoglobin binds an oxygen, it will become more. It'll have a higher affinity for an additional oxygen, and every time hemoglobin gives up an oxygen, it'll have a lower affinity for the oxygens that remain. This allows it to pick up the maximum amount of oxygen in your lungs, because every additional oxygen will make it pick up even more, and it also maximizes the oxygen that it gives off in your muscles or other locations that are using the oxygen, because it will, every time it gives off the oxygen, it'll give off even more. And so that's why cooperativity is important. Now, our last type of regulation is feedback inhibition. Now, feedback inhibition isn't inherently different from the rest of them. It's just the way that the rest of those functions are used. In feedback inhibition, the end product of a metabolic pathway is actually the inhibitor for an enzyme earlier on in the pathway. So you can kind of see in this diagram, you start with the red molecule, a reaction changes it to the orange molecule, and then another reaction changes it to the green molecule, and then the last reaction changes it to the blue molecule, which actually acts as the inhibitor for that first enzyme. By using feedback inhibition, the cell prevents itself from making too much of this product. If there's so much of this product left over in the cell that it will bind to this enzyme rather than being used by the cell doesn't want to produce any more of it. And because it's preventing this enzyme from functioning, it's preventing the cell from wasting this starting molecule, which could be valuable in another pathway and producing another product. And so that's really it for enzymes. They're as simple as that.